In this video, I will be reviewing all the material you'll need to know for the MCAT's content category 2B, which deals with microbiology, its history, its biological context, and its various compositions. I'll start with cell theory. The first man you're going to want to know is Robert Hooke. Robert Hooke was one of the very first microbiologists. He ground his own lens, which are the precursor to today's microscopes. He wanted to know, what does cork look like when I magnify it under my lenses? So, he saw these tiny little structures that resembled the cells that the monks lived in during the time. Now today we know these tiny little cell-like structures are really dead cell walls. And he is famous for giving cells their name. Now the second man you're going to want to know is a man by the name of Anton von Leeuwenhoek. He is also one of the very first microbiologists. He made his own lenses, and he wanted to see what is in the water sources around where he had lived. So he took a sample of water from the local river, and he magnified it, and he saw all these strange little animal-like creatures just swimming about. And these types of creatures have three basic shapes that you'll need to know for the MCAT. There are also shapes such as fibrous and star-like shape that will not be on the MCAT. Now the first shape is Bacillus, which is a rod-shaped bacteria. Now the second is the Coccus, or for the plural version, Coccus. Now this is a spherical shaped bacteria. And lastly, the Spirilli. This is the spiral shaped bacteria. Now these three shapes are what you need to know for the bacteria shapes on the MCAT. Now the Bacillus is usually drawn as a little Twinkie shaped rod with a flagella on the end. Now, the coccus, two main examples would be, say, Streptococcus, which is a chain of spheres, is known for strep throat, and Staphylococcus, which is a grape-like structure and is known for staph infections. And spirochetes are pretty much known for being spiral-shaped. Now, Bacillus subtilis is the main gram-positive organism studied by microbiologists, is perhaps the second most important to research, second to only E. coli. I remember Bacillus shape because it looks like a baseball bat, and it has the BA at the beginning, and that's how I learned the shape of Bacillus, because it's shaped like a bat and has that ba sound at the beginning. I remembered Coccus and Coccus because it kind of sounds like coconut, and it also has the same shape. The COC at the beginning of Coccus and Coccus and coconut kind of helped me to remember that's a spherical shape, or shaped like a basketball. Spirochetes, spirals, Spirilli, they all had the spur at the beginning, and that's how I remembered that as well. So I kind of remembered it based on how their words were pronounced and structured. Now, Loiois Pasteur is one of the most famous biologists of all time, famous for the pasteurization process. Well, he disproved the theory of abiogenesis, where life comes from non-life. So he developed this apparatus that prevents contamination, and he put all the materials you need to form cells, and he heat killed anything in his solution. So he had no life living, but he had all the basic components to make up these life forms. So he wanted to know that with the building blocks, would they spontaneously combine and start making new cells from just the basic building blocks. And he found out that wasn't the case. So the conclusion was that cells only come from pre-existing cells. Now, you're going to need to know as well the prokaryotic domains on the MCAT. There's two: the archea, known for being extreme, and a bacteria. Now, these are the two prokaryotic domains that are tested on the MCAT. The archea are extremophiles, and they are located in, say, extremely hot places or extremely cold places like Arctic conditions or spas. These sorts of extreme temperature archaea are known as thermophiles. There's also salty, say in the Dead Sea, or high acidic or high basic sorts of environments, and they're usually found there as well. Now, methanogens are a special type in which they line the guts of cows and they produce methane gas. Bacteria are known for lacking membrane-bound organelles. Examples of membrane-bound organelles are, say, mitochondria. There's a thin cell membrane surrounding the energy production apparatus in the mitochondria. And while they may have things like ribosomes, they won't have, say, an endoplasmic reticulum, which is what differentiates them from our eukaryotic cells. Now, another important distinction between, say, bacteria and us would be the cell wall. Now, all our cells have cell membranes, and bacteria also have cell membranes, but they also have a cell wall. Now, plants also have cell walls as far as eukaryotes go, but we do not have the cell walls, which is an important distinction between prokaryotes and eukaryotes. So the takeaway here is that the bacteria will have the cell wall and cell membrane, and that's important for, say, medication that specifically disrupts the cell wall. Now, bacteria also have an apparatus known as a flagella. The flagella is a way in which bacteria can move about in a suspension. It can propel them forward, or it can tumble, and that allows them to change direction. Well, at the base of the flagella is this biological motor that spins the flagella in a whip-like fashion to propel it forward, or if it, say, reverses the direction, it can tumble left and right in order to change direction. The flagella is an important motor for getting around for prokaryotic organisms, along with, say, cilia. Flagella is usually the big one. Now, bacteria produce by binary fission, where one cell becomes two cells. And if you can imagine, it's kind of like having a water droplet where you kind of just pull the water droplet apart, say by toothpicks, and you'll kind of see it divide in the middle as it kind of clings to itself. Well, bacteria will be separating one cell into two cells and splitting evenly the genetic material, so it'll essentially be two copies of cells. Now let me draw this a little better. They have this kind of little cleft in the process of the division, but you'll see the genetic material, my little circles are kind of genetic material in general, and they'll form a cleft. And that's the takeaway from binary fission.